Cut off a salamander's leg and it grows right back. Cut off a mammal's limb and the body rushes to seal the wound, leaving behind a thick scar. Evolution favored this trade-off, prioritizing rapid wound healing to survive predators over the slow luxury of perfect limb regeneration. But that rule of permanent scarring has a bizarre exception hidden at the very end of our hands. In both mice and humans, the extreme tip of the finger can actually regenerate. If a mouse loses the tip of its digit, just below the nail, the bone, skin, and surrounding tissues regrow perfectly, restoring the finger to its exact original shape. This diagram maps out the boundaries of that exception. Cut at the blue line, near the nail, and the digit regenerates. But an amputation just a few millimeters lower, at the gray line near the knuckle, inevitably results in a permanent scar. Two nearly identical injuries on the exact same finger produce completely opposite biological outcomes. Understanding why the tip regrows while the knuckle scars provides the template for unlocking total limb regeneration. For decades, efforts to crack the code of regeneration focused almost entirely on biology software, the cells themselves and the genetic pathways inside them. The cells found in the regenerating fingertip and the scarring knuckle are essentially identical. It is their immediate environment that makes the decision to heal or scar. This environment is the extracellular matrix, or ECM. It functions as an active director, sending mechanical and chemical signals that control exactly how cells behave. Since the ECM issues the orders, triggering regrowth doesn't necessarily require engineering the cells. It requires changing the physical architecture of the matrix those cells live in. We can see this architecture in action by looking closely at the microscopic environment of the knuckle wound that fails to regenerate. The matrix surrounding a knuckle amputation is dominated by dense, highly organized bundles of collagen, creating a rigid wall that boxes the cells in. At the tip of the regenerating fingertip, the physical space feels entirely different. This zone of regrowth is known as the blastema. The blastema is saturated with hyaluronic acid, or HA, creating a soft, fluid, and viscous environment. The hyaluronic acid protects the cells by physically pushing incoming collagen fibers apart, blocking them from assembling into a stiff, rigid grid. This physical tension acts as a mechanical switch. When cells feel a stiff collagen matrix, they begin depositing scar tissue. When they feel the soft, fluid pressure of an HA matrix, they relax, multiply, and rebuild bone. Hyaluronic acid serves as a physical prerequisite for repair, forcing tissue into a state of regeneration by actively blocking fibrosis. Testing the link between HA and regeneration meant removing it from the one place it normally thrives. Following an amputation at the tip of the mouse digit, scientists repeatedly injected the wound with hyaluronidase, an enzyme that destroys hyaluronic acid. Without the protective barrier of the HA molecules, the matrix immediately lost its softness. Stiff collagen fibers moved in and linked together. These images from the study capture the impact of that stiffening matrix. While the top photo shows normal digit regrowth, the bottom photo shows a stunted, failed injury that scarred over once the softness was removed. Without the mechanical softness provided by hyaluronic acid, mammalian limb regrowth appears to be impossible. If biological softness acts as the switch, it raises a new possibility: hacking a scarring knuckles matrix to make it regrow like a fingertip. To test this, researchers used a component of the extracellular matrix called HAPLN1. HAPLN1 is a link protein that acts as a molecular anchor, tethering hyaluronic acid networks together so the soft molecules don't degrade or drift away. The scientists performed a knuckle-level amputation, which normally scars, and introduced cells designed to overproduce the HAPLN1 protein into the wound. The HAPLN1 successfully locked the hyaluronic acid in place, keeping the environment soft and blocking the scar. For the first time, this allowed actual bone regrowth to occur in the knuckle. By artificially stabilizing the physical tension of the matrix, the researchers successfully coaxed a non-regenerative wound into repairing itself. For decades, regenerative medicine has focused on stem cell therapies and genetic manipulation. Reversing amputations might instead require retuning the physical mattress the cells sleep on. We now know that the true capacity for complex limb regeneration lives in the dynamic, fluid space between our cells.